What's up, TUMNAD lectures? In this video, we're going to be talking about the sympathetic nervous system part 2. If you have not yet watched part 1, we talked about part 1 whereby we saw that the sympathetic effects on the head and the neck through what we call this kind of the carotid plexus, and then even also sympathetic effect to the esophagus, and then the lungs, and even also the heart through what we call the cardiopulmonary plexus. We talked about all that and where they are going to be coming out from what we call the thoracolumbar region. Okay. Now, we talked about that for the case of what we call the carotid plexus, we saw that it was T1, T2, and T3. The preganglial motor fibers were coming out without synapsing in the chain ganglia, and then they are passing through the cervical ganglia without synapsing, and they are synapsing on what we call the superior cervical ganglia, and then from there the postganglion motor fibers were coming out to coil around the internal carotid artery, from where we're going to be having what we call the postganglion motor fibers going to innervate different structures of the head and the neck. We looked about all that, and then we also said that we're going to be having the preganglion motor fibers coming from T1, T2, T3, T4, and T5 coming outside up through the chain ganglia, and then the postganglion motor fibers coming out to make this kind of the union, and even also with the combination from the cervical ganglia, making what we call the cardiopulmonary plexus, which was going to supply different structures within the chest, like the lungs, the heart, and even also some part of what we call the esophagus. We looked about all that. So now in today's video, we're going to be seeing how the sympathetic effects going to be causing different changes within the visceral, primarily the GIT. So let's go ahead and get started. Now we are going to be having the preganglion motor fibers coming from T5, T6, T7, T8, and T9. They come out from the thoracolumbar region. They don't synapse through the chain ganglia. Okay, they are going to be passing without synapsing. And of course, whenever these nerves come out without synapsing, how do we call that? It becomes now a splanchnic nerve. Okay, so now we have T5, T6, T7, T8, T9 coming without synapsing, and then they come and make this kind of union, and this kind of preganglion motor fibers, they are what we call the greater splanchnic, the greater splanchnic nerve. Okay, the greater splanchnic nerve. So now we're going to be having these kind of preganglion motor fibers coming, and when they come, they're going to be going to different parts within the viscera. So now you're going to be having the first branch coming and then synapse with this ganglion. You see this ganglion? So now this ganglion here is what we call the celiac ganglion. Okay, is what we call the celiac ganglion. And of course, now you're going to be having the postganglion motor fibers coming out as what we call the celiac plexus, going to innervate different structures within the viscera. So now you're going to be having now the postganglionic motor fibers coming, and then we shall have some of them which are going to go and innervate what we call the stomach. Shall have some of them coming to innervate also what we call the biliary system. Okay, we shall also have also what we call those one which are going to be innervating what we call the pancreas. Okay, so now we need to see what is going to be now the sympathetic effect to these structures of the GIT. Okay, so now what's going to be the sympathetic effect to what we call the stomach? Okay, you know the stomach is going to be having a lot of changes whenever sympathetic nervous system is activated. Okay, so now starting is what we call the blood flow to the GIT. Okay, now whenever we have sympathetic nervous system activated, here we don't need something like we want to slow down the digestion, because at this time we don't need a lot of digestion. We need to fly away, or even to get more energy to fight back. Okay, so now in that way we are going to be slowing out things concerning GIT secretions, things concerning blood flow to the GIT, things concerning peristalsis. We're going to be having all of them getting slowed down. Okay, so now what we call, you're going to be having a decrease. You're going to be having a decrease in blood flow to the GIT. Okay, blood flow to the GIT is going to be slowing down. Okay, then that peristaltic movement within the GIT is going to be slowed down. Okay, in the stomach, so have what we call a decrease in what we call peristalsis. Okay, a decrease in what we call peristalsis. Okay, and then how about if we decrease the blood flow to the GIT, especially the stomach? How is going to be the effect on the absorption? Because in the stomach we have some drugs which gets absorbed in what we call the stomach. So now it means you're going to be having reduced absorption. Okay, so you're going to be having what you call reduced absorption. Okay, what we call reduced absorption. So now, how about whenever we put in some foods in the GI, there is always some kind of the gastric secretions. But during this state of stressful moment, they are chasing us. We are going to be having a decrease in what we call gastric secretion. Okay, so you're going to be having what we call a decrease. Are going to be having what we call a decrease in what we call the gastric secretions. Okay, the gastric secretions. And of course, the gastric secretions, they are quite very many. We have the enzymes, we have what we call the acids, we have what we call, and even all other, the pepsin, all those kind of secretions are going to be reduced whenever sympathetic nervous system is activated. Now, how about with these kind of the sphincters? We have this pyloric sphincter and even also have what we call the lower esophageal sphincter, or which you may even call what we call the cardiac sphincter. What's going to be the effect, the sympathetic effect on this kind of these structures? You're going to be having what we call constriction. Okay, we're going to be having what we call constriction. We're going to have what we call constriction. Why? Because now we don't need the chime to come from this stomach to come to the dam, okay? And even also we don't want anything. For example, if you're running away, there's that kind of the shaking of the stomach. We don't want some kind of this gastric content even again to flash back. It will cause what we call the gastric reflux, okay, which is very, very, very dangerous, going to be damaging what we call the different cells line, what we call the esophagus, okay? We don't want anything to be coming here, and we don't want anything to be leaving in the stomach. So now that's why these kind of sphincters, they have to constrict, okay? It's not such another good thing. But now you have to be in mind that the neurotransmitter which is going to be released, there's going to be no epinephrine, okay? So now, what's going to be the effect on what we call the liver? Of course, you know, in this stressful moment, we need a lot of the glucose, and the liver is one of the main storage center of what we call the glucose. So now, because the digestion is going to be slowed down, absorption and everything. So now we need the liver to give us more glucose, okay, from what we call the stores of the glycogen, meaning that we need more of the glycogen to get converted into the glucose. And how do we call this process? This process is what we call glycogenolysis. Okay, you're going to be having what we call glycogenolysis. Okay, that's going to be the primary effect of the sympathetic nervous system on the liver. We need more glucose. Okay. How about the biliary system? Because the biliary system is going to be providing us the bile, which are going to be aiding in emulsification of fats. But because now the digestion is slowed down, we don't need more of the biliary secretions. Okay, so now you're going to be having reduced biliary secretions. Okay, you're going to be having what we call reduced biliary secretion. Okay, we're going to be having reduced biliary secretion because we will be having a lot of substances coming from the biliary system. We don't want them at this time because we want to get a lot of the energy that we can run away. We don't want to digest at this moment. Okay, that's why we have to reduce the biliary secretions. So now, what's going to be the effect on what we call the pancreas? You know, the pancreas is going to be having two kinds of the tissues. There is this kind of the tissue which is going to be constituting the bigger percentage, even approximately 90%. And then we also have those kind of the small tissues which are going to be very, very, very few. See this kind of these red kind of these tissues, these red tissues which are very, very, very few. These kind of these tissues are what we call islets of Langerhans. Okay, islets of the Langerhans. Now, this tissue is the tissue which serves what we call the endocrine role. Okay, it's going to be serving what we call endocrine role. And when we talk about endocrine role, we mean we're going to be releasing hormones direct into the blood. Okay, so now we want to first zoom out to this kind of the tissue. We zoom it and see the kind of the cells which are going to be residing in this kind of cells, or this kind of the tissue we are calling the islet of the Langerhans. You see this kind of this cell here. This cell here, we want to call it to be the beta cell. And then this cell here, what to call the alpha cell. And of course, you're going to be having a quite number of the cells in the islet of the Langerhans, like of the epsilon, the delta, but I don't want to focus on those ones because they are not so much significant in this way, but we want to see what do these cells, what we call the beta cells, produce and the alpha cells produce. You have to know that the beta cells produce what we call insulin, and then the alpha cells, they produce what we call glucagon. So now, in this kind of sympathetic effect, do we need more insulin, or we need to inhibit what we call the beta cells? Because insulin is going to be taking now, is going to be making all the glucose to be reduced in our circulation. Okay, that means we need to now to block the production of insulin by these beta cells. Okay, so now what's going to be the sympathetic effects? We're going to be having inhibition. We're going to be having inhibition. Okay, inhibition of what we call the beta cells. And what about for the example of the alpha cells? We need glucagon because we want to raise the levels of glucose in this kind of distressful moment. So now you're going to be now stimulating. You're going to be stimulating. Okay, you're going to be stimulating. You're going to be stimulating the alpha cells to release more of the glucagon, which will aim at increasing the glucose levels, which is so much needed in this kind of distressful moment. Okay. Now I will also have these kind of these very many cells. See these black cells, these black tissues. This one's going to be constituting the bigger percentage and going to be serving what we call the exocrine role. Okay, and when we talk about the exocrine role, we mean it's going to be producing its secretion direct into the ducts. Okay, so now the whole of this tissue, you're going to be first cutting it out. Okay, we're going to be cutting it out. We zoom it and see which kind of tissue is this. Now, this kind of the tissue is what we call the acini. Okay, is what we call the acini, and this is the one which is going to be responsible for production of what we call pancreatic digestive juices, which are going to be aiding in what we call the chemical digestion of food in the what we call the small intestine. Now, in this kind of the stressful moment, do we need more of this chemical digestion? No, that means you're going to be inhibiting this kind of the acini cells from producing what we call the pancreatic digestive juice. So you're going to be inhibiting. Okay, we're going to be inhibiting. Okay, we are going to be inhibiting the activity of the acini cells from producing the pancreatic digestive juice, because at this time we don't need to digest. Okay, because at this time we don't need the production of the pancreatic digestive juices because of the slow down kind of the digestion. Okay. So now that's going to be now the effect of this kind of the postganglion motor fibers on this kind of these structures. Okay. Now we are going to be having again these kind of fibers which are going to go to this kind of this structure. You see this kind of this structure here. We are going to be having what we call the preganglionic motor fibers. Okay, these kind of preganglionic motor fibers are going to be coming and then they terminate inside this kind of this structure. You see this kind of this structure which is just going to be above this kidney. This structure is what we call the adrenal gland. And of course, the adrenal gland is going to be having two kind of the portions, the outer one which we call the adrenal cortex, and then the deeper structure which we call the adrenal medulla. Now, these preganglionic motor fibers are going to be moving all along and then they terminate in what we call the adrenal medulla, primarily on what we call the chromaffin cells. And of course, the chromaffin cells are going to be modified postganglionic motor neurons. Okay, this is going to be now an example of what we call the intramural ganglion. Okay, so now these preganglionic motor fibers, they are going to be now releasing acetylcholine, which will bind on what we call the nicotinic receptors on what we call the chromaffin cells, which will stimulate them and release these two kind of chemicals. What are these two chemicals? The first one is going to be norepinephrine, and another one is going to be what we call epinephrine. Okay, and these two chemicals are going to be entering into what we call the blood, and then they go and act on what we call the adrenergic receptors all over the body where they are, and leads into what we call a different kind of physiological process. Now, for example, epinephrine, if it goes to the, for example, the cardiovascular system, is going to be raising what we call the heart rate and even also the strength of the contractility. And if it goes to the respiratory system, it's going to be causing a bronchodilation and even all that. Okay, so now this is going to be now the preganglionic motor fibers stimulating the chromaffin cell to release epinephrine and then norepinephrine. Okay, it is also such another beautiful thing. What else? We also have this kind of preganglionic motor fibers. We still have this kind of preganglionic motor fibers which are going to be coming, and then these fibers are going to be now